This is blood sample.
We didn't use all of it?
No.
Sweet. You said 200 milliliters?
About 200. I might try 150 first, because what matters is the size of the pellet. So it matters how much blood to ethanol ratio we have. And so it's kind of a guessing game. But lately, when I've been doing it, it's about 150 for the type of, for how much blood is in this sample. 150 microliters of blood ethanol.
And what else do we have out right now?
This is just the lysis buffer. It's called a TESR buffer.
And did you made that up ahead of time as a master mix?
Yes.
Okay.
And then we'll add the RNase A right before we use it. So this doesn't have RNase A in it yet.
Okay.
So what we're going to do, and Emily, I'll have you do this on one of the tubes here. We're just going to go and try and pull from the bottom so we have the most tissue possible. But see, it's like not sucking up. And there it goes. Because there's going to be lots of tissue that it's trying to pull up. And then that's going to go there.
See Skylar
not being able to work a freaking centrifuge. Okay, so now we're going to spin this.
So this is a slow spin.
Really slow spin. Like, I believe the protocol says like 1.5.
Okay.
And so we're going to do that for 3 minutes. So you can't even hear the centrifuge going right now. The reason we want to keep it at a slow spin is we don't want the red blood cells to lyse. Because if those red blood cells lyse before they get into the lysis buffer, then the DNA is exposed to RNase and DNase and everything else.
So that was just to get the blood down to the bottom.
That's just to get the blood to go down. So that's about the size of the pellet we want. Okay.
So now you're removing the supernatant.
We're just removing the ethanol out of the supernatant here. So what I'm going to do is be very careful. Open it. We don't want to disturb that pellet. So what I'm going to do is start from the top and just slowly come off so I'm not losing any of that blood cells. And then work my way down. And then that we can just put there. And then keep removing. I'm going to get a little blood there at the bottom. But then that's good to go. Most of the ethanol's gone and we just have our blood cell there. So I might take a P20 now and try and get that last tiny bit. We're going to see that last little bit there at the crease.
Like this is kept at the -20. Okay.
Yeah, I just have it in the fridge to keep it going. So I'll have you go ahead and just pipette it. So this is already set to 14.
So those 4 purple lids are our 4 samples moving forward.
Yes.
Yeah, so those have the TESR buffer in them.
Cool.
But the R in that is the RNase A reaction. Right before we activation.
Yeah, go ahead. And just invert them once. Cool. So now this is good to go. So what I'm going to do, we have our blood sample here. We're going to just come in here, suck up some of our buffer. No, I don't go quite all the way to the top. Then we're just going to rinse, get that blood resuspended.
So this is a transfer step.
Yep, and this is just getting everything like that to go over here. So that's good to go. And sometimes it takes more than one step to do that. But there's no more blood in the tube there. So then if you look at this, you can see it's sitting on the top and it's already starting to separate and lyse. So we're just going to invert that. You can see it already getting stringy and jelly. That's already lysing the cells open. And so the RNase A that we added is what's degrading all the RNA in there, and then we're going to add proteinase K in an hour, and that'll destroy all the proteins in there.
Okay.
There's a blood sample.
Okay. So there's still some of that. We didn't use all of it.
No. You don't want to shake these up. There's SDS in it, which is the detergent, and it'll make it really bubbly. Just a slow inversion a couple times.
Did you set a timer, like a strict timer? Every 20 minutes you go in?
I don't set a strict timer. It's just kind of normally I'm doing other stuff when I'm doing this, and so whenever I think about it, I just go and do it. Give it another couple good mixes, and then so again 37 because that's when RNase A is going to be most active.
Okay.
Proteinase K is active at 37 obviously because it's an enzyme, but it is also active at 50 and that'll help break everything else down.
Proteinase K. So we keep this in the fridge until we're about to use it?
This is always in the fridge. So this is just in water, and so normally like the cold stuff is going to pipe out a little different if it's more volatile or something like that. But this is just proteinase K.
Ah, okay, got it.
This is just the prep steps up here. So we have our water bath going. We obviously prepared our TESR buffer.
How long is the incubation step?
This is an hour-long incubation step.
Okay.
We need 30 milliliters of 70% ethanol. So we need 120 because we have 4 samples. And then put that in the freezer. So put that in the -20 here.
Okay.
Then I need you to spin our phase lock tubes. I need you to spin each of those at 1500 G for 2 minutes. And so all of our spin steps are going to be in the fridge today. We have the big centrifuge in here, so the 15 mil things fit in these holders here. This is what the phase lock tubes, this gel part, which should be at the very bottom, this is an old tube that already did an extraction on. So what that first extraction step is going to look like is this here, where we have this bottom part is going to be protein and other cellular components that we don't want, and the top part is going to be the liquid phase that we keep moving on that's going to contain our DNA.
Okay.
Then this top part we can just pour off. We never use a pipette the entire time. These have phenol and chloroform, which can cause cancer. So once you guys get those pre-spun, leave them in the fridge.
So we just finished the hour-long incubation, and we're adding 50 microliters of proteinase K. And then the next step is another incubation at 50 degrees.
So this is 3 hours or longer. So if it goes 4 or 5 hours, then we're fine.
Okay.
It just means we're going to be here longer tonight.
Okay.
The time steps are not as
They're not
crucial.
Not as crucial.
It's just the order, I guess.
Yeah. So the time steps are not crucial. Like the volumes, obviously you want to get pretty close to them, but it's not like you have to be 100% exact on this protocol.
Okay.
And then yeah, this step is going to take us probably like 2 hours here because you can see all the 10-minute wait steps. Because this hook has to fit inside of a 1.5 mil tube.
I thought,
No,
I can't remember what we did final one.
we don't want a complete loop. We want it a hook.
Oh, I have it.
There it is. Woo. Okay.
Yeah, there you go.
There we go. But then once this is done, I just set them here to cool, and then we can stack them in here.
Cool.
See that still? No hole there. And then what we're gonna do get our way in, and
Oh, I see. Okay.
voila,
So let the weight.
it's a hook.
Is that a good looking hook?
This is decent, yeah.
Okay.
You can see it's starting to close up. Alright. So then if you look at the very tip, you can see there's no hole, right? It's completely rounded. So that's good to go. Then we go just a tiny bit in. As soon as it starts to fall, kind of lift it up and then spin it. And we'll stick them in and cover them up with the rest. They look really good and digested.
So that one doesn't look as good?
Well, it just had a lot more tissue in it.
Gotcha.
We've broken down the RNA that came out of the live cell. We've broken down the protein.
Okay.
Let's get a few chairs over here, maybe.
This step's really easy. You're just going to take your cap off. You're going to take this cap off. And there's all that soap I was telling you guys about. And then everything from here on out is just pouring. So you can see that goop just go in. And then you want to make sure to get everything possible. So come down here and kind of just tap it to make sure you get those last drops. Cap that. Easy peasy. Nice and suck it up, ease blowing it out. So what you're going to do is push that bolt while holding either S or A. It'll get the air out of the bolt, so then you can suck it up. You can see that sample's already going to start clearing up. And after this first extraction, a lot of that cloud is going to be under here.
So you just use regular tape?
Yeah, I'm just using tape just to get them so they don't move around on the tray. It's just going to sit there and shake, gently shake our samples for 10 minutes.
At 23 is the speed.
23 is the speed, and the tilt is as far as it'll go.
We just finished the 10 minutes of rocking, and now we're doing 10 minutes at 8,000 G in the 4 degrees.
So the reason we do everything cold is because we're pretty sure that the chloroform heated up a little bit, and so we keep everything cool because it's going to slow that reaction down. But even with these fancy, expensive tubes, they get a little soft. So like, that's why I keep everything cold. Okay. My sample looks like beforehand, right? We're going to take number 2. So it's not the top or the bottom. So it's like the middle, like, I feel like that's the best way. So then I'm going to slowly pour I'll take the one. Well, you should see how many samples I have. Okay. 5 milliliters. Tight. And then you can see it's starting to separate a little bit again. And that's why I put on the off.
What number are we on?
We are on 11 right now.
Cool. So now we're moving to 3 ml of chloroform moving forward.
Okay. Gunners is really clear right here. Both the top and the bottom are clear. You can see the blood samples are starting to clear up a little bit, but they're still getting rid of things.
Yeah.
And then obviously this sample still looks like shit.
So that's what a shit sample looks like.
But there's still probably DNA in here.
Yeah.
So this is still good, but I'm worried about Gunners at this point.
So this step is the same as the other ones?
Yep, exact same. Just chloroform this time instead of phenol chloroform.
I have an idea. Why don't you put the chloroform in the old tube and then...
But that's the only difference between this step and the previous one.
Okay, we're on our last one. And then while we're doing these 2 10-minute waits, we're going to set some stuff up under the hood. One update that we're trying this time is instead of adding the 3 ml to the new tube, I'm trying to put it into the old tube and use it as a wash and then dump that into the new tube, and it seems like it's carrying over DNA. You know what that bubble was, or the ball I was telling you? You know how you said the water doesn't dissolve into fluid? Just plug it from that state. So just make sure the caps are on. Just been taping it on this thing.
So we're going to pour that top piece. We're going to do the salt precipitation step, swirl mix. Then I'm going to carefully layer my ethanol, and it's going to form 2 layers, and you're going to kind of slowly, gentle, like rotate this back and forth. Ideally, what you're going to get is a ball of DNA. That ball of DNA you're then going to hook out. Then we're going to move to the next sample. And so to get prepared for this step, what we need to do is we need to get cold ethanol in the 3 backfilled tubes. Because all it is a wash step. You're just going to dunk your hook in there and let it sit. And then there's nothing that it actually does for the sample. And so about 5 is fine. Just eyeball 5.
Can you say like which numbers?
We're doing 18 to 22 in 1 lump per sample. Does that make?
Okay.
So I'm going to take 1 sample and I'm going to precipitate it with the salt, then the ethanol. I'm going to hook it out, and then Gunnar, you're going to start and do the same thing.
Is it time sensitive?
So I don't know if it is, but I know, like, once that salt and you get that first round of like swirlies, or that you see the salt mixed in, I think that part it's kind of important so the salt doesn't start to diffuse a little bit more and it stays bound to the DNA. I think that part is important to then quickly add the cola ethanol.
for the wash step is you pick the hook directly up and then you put it into the next 50 ml conical that has 70% ethanol in it, and then leave it for 5 minutes, do it again. And that's the end of today.
And I'm just going to tap it on the side to where I get most of the ethanol off. So I'm going to tap both sides of the hook, or all sides of the hook, to try and get as much ethanol off as possible. And then it's going to go in here, and it's going to sit for like 4-ish minutes to dry off, and then we're going to add the tris to it. Then Emily, as soon as he's done, go ahead and switch to your last wash. And do the same thing to tap your hook off. So dry, we're not getting any residue on the side when we touch the glass to the side of the tube.
10 millimolar tris.
So then our hook's now covered completely in there. So now we're going to break the hook off. So I'm going to lift it up out of the bottom a little bit, put my thumb right there, and break it off inside. So to break the hook off, I'm going to lift it up about halfway out, put my thumb against it, my thumbnail, and then I'm going to bend towards it. So it broke off a little higher than I wanted. Let's see if it's going to fit. Nope. So I'm going to try it again. Again, break towards me. And then I can now close my